Okay, so let's start. So now we'll start the afternoon session. So Professor Mauro will give his first lecture now, and then the second part will be tomorrow morning, okay? So it is my great pleasure to introduce Professor Mauro Copelli. Mauro is a professor of physics at Federal University of Pernambuco. Born and raised in São Paulo, he earned a physics degree from the University of São Paulo and a PhD in physics at Limburg's Università Centrum.
There you
go. More or less, yeah, I think.
Good Dutch.
Under Christian van den Broek. He later pursued postdoctoral work at UCSD and later at the Universidade Federal Fluminense. His research focused on critical phenomena in neuroscience using tools from statistical physics and experimental data. More recently, Mauro has made important contributions to computational psychiatry. As a side note, Mauro told me that although he was born in São Paulo, he's now fond enough of Pernambuco to appreciate the song of Madeiro do Rosarinho, a traditional music from the region.
There you go.
Without further ado, please, the floor is yours.
Thank you. Thank you. I want to thank Danielle and the other organizers for the invitation to be here. I've been to these schools at ICTP-SAIFR for a long time now. It's always a pleasure. It's really smart people in the audience, good questions, so I love it all. Please feel free to ask me questions whenever you want to, okay? So, this thing I'm going to present I gathered this name, Collective Neuronal Behavior and Critical Brain Hypothesis. We'll be divided into lectures. So today I wanted to give you this idea of what this is all about, the notion of criticality in the brain, a simple example of a toy model that we introduced like almost 20 years ago, and neuronal avalanches, which is a concept which has been around also for more than 20 years now, and how these things have guided this field of research. And my goal, perhaps too ambitious, is to tell the story of this field, which is a small field, very small field in neuroscience, from 20 years ago up to today, and seeing how techniques evolved from the experimental point of view a little bit, because I'm not experimentalist per se, and also from the theoretical point of view, the tools that people have been developing to understand these things, okay? So the general context in which I'm going to introduce these ideas, remember, I used to be a theoretical physicist, okay, like some of you guys. Today, I don't know what I am. And there's this very, very, very panoramic question, like, do we have a theory for how the brain works, like a good theory? I mean, if I ask, please, close your eyes and think of a blue elephant, okay? Now, everybody can do that. So what the hell happened right there when you did this? So how did this concept of the elephant and the blue came together and an image appeared in your head? So we don't have a good theory for that, right? When I say we have a good theory, we need a scale for good theory. Let's take physics, theoretical physics. We're here, we can speak about physics here. So, right? So I see physics like this well-groomed golf course where things have been well taken care of for 300 years, and people go to Switzerland and take anti-proton, anti-electron, make an anti-hydrogen, right? Hydrogen of antimatter, measure the spectrum and see if it fits the normal hydrogen, because it must, according to theory. If it doesn't, everything falls down. It's very sophisticated, right? And by the way, it does, okay? Actually, the last experiment in this setup was to do everything again in a vertical tube to check whether it falls. And it does fall, as it should. So it's very sophisticated theoretical building, so to speak. Of course, there are some issues like 80% of the matter of the universe, apparently we don't know what it is. And so there are issues, of course. But still, we have some kind of theory. Compared to that, I think that theoretical neuroscience is something like this, going to the woods with a blunt knife and cutting branches and discovering fantastic things just by cutting branches. So this is not a criticism of neuroscience, it's just a recognition of the maturity of the science from a theoretical point of view. So this, I love hiking, I don't know about you, so there's plenty of opportunities to think and come up with new ideas And now there's a bunch of data coming out, the public available data, right? So, so it is in this context that I'm going to mention the things I'm going to mention today about brain criticality, in the sense that it's just a hunch of some people, some idea, it's a hypothesis, a conjecture, that people thought might be useful in such a context of kind of a lack of major fundamental principles to guide, theoretically, what's going on in the brain. Okay? So, so what is this thing about brain criticality? Let me start with some experimental data that kind of gave rise to the boom of this. It's a small boom, but it's still a boom in this field, which came out in 2003 in Dietmar Plain's lab at the NIH. So they take slices of red cortex plus a little bit of VTA, and they put they had these 60 electrodes and measure local field potential, which is kind of average electrical activity around electrode, and they measure this spontaneous activity here. This is time, these are electrodes. They have some kind of silence, and then everybody more or less together, like show some activity, and then you have silence again, and then some activity again. It looks like it's synchronized, but it's actually not. If you look in detail, this looks synchronized, it's a larger time scale here. It looks synchronized, but if you look carefully, it's like a spatial temporal pattern, which then is followed by silence. So they call this activity here between two silences a neuronal avalanche, in the sense that there's a separation of time scale. The duration of this event is much less than the recurrence interval, like an avalanche of, I don't know, snow, right? It has to snow for a long time, then accumulates, and then poof, there's some relaxation process, which is much faster than the accumulation time. And if you choose any reasonable measure of what the size of this avalanche would be, for instance, you take this activity here, you take super threshold activity above, I don't know, 2 standard deviations of the mean or something like that, you call it an event, and you count the number of events. So you could ask the question, okay, so here we have 1, 2, 3, 4, 5, I don't know, 27. The size of, this is an avalanche of size 27 and the duration of 3 time beams, okay? And you have another event, and another event, and another event, and then you calculate the statistics of this, you gather a lot of data. This is an in vitro experiment, so it lasts many, many hours, very stable. And you do the statistics. So what is the probability of finding an avalanche of size S in this context, in this experiment? And the answer is that this is a probability. So this is a log scale. This is also a log scale. You have this very beautiful, actually, power law. Of course, not many decades, because this is limited by the number of electrodes. And it's a beautiful power law. Actually, it's very well described by this exponent here. And your natural instinct should be, so what, right? Who cares? Well, actually, the fact that it is a power law, of course, with a cutoff here, means that it is a fat tail distribution. And what does that mean? It means that questions such as, what is the mean size of an avalanche? What is the typical size of an avalanche? What is the mean plus or minus standard deviation of this process, of this observable that I'm decided to look at, the size of an avalanche, like how much activity there was? All these questions are meaningless, because if you calculate the standard deviation of this thing, of this distribution is infinite, right? So that means that your system is governed by fluctuations. It doesn't have a typical size. Actually, it's scale invariant, so we don't have a typical. And so this raised the ears of many physicists who, when they saw that, they recognize this as a signature of what could be a critical process. I'm going to come back to that. This, the fancy name for this is non-trivial scale invariant statistics for this kind of distribution here. The distribution for duration is less beautiful, but this slope here is -2. And then later, in later years, the same group developed the same kind of, obtained the same kind of results for less reduced preparations. So instead of a slice, they had like, this is an anesthetized rat, again with the same slope here, the same exponent, 3/2. And later on, with unanesthetized but resting monkeys, so they're measuring premotor cortex of the monkey, and they measure, again, the same exponent, always with local field potentials. Right. So this field, these ideas that were circling around for some time, the critical brain hypothesis, which is the following. This is the idea that the brain, regarded as a dynamical system, very high dimensional dynamical system, operates near a critical point. What is a critical point? The critical point is sketched here in this cartoon, in which you have some order parameter of a physical system. This is a concept borrowed from physics, okay? This is a physical system. You have some order parameter and some control parameter, and nothing happens as you change the control parameter here. Nothing happens, nothing happens, nothing happens, nothing happens, until you reach a critical point, at which you have a qualitative change in the system, a macroscopic qualitative change in the system, which then changes, and changes continuously here. So this is a picture. I'm going to give us an example, a physical example right now. And this idea has been around for some time. So we have this physicist called Per Bach who introduced the idea of self-organized criticality back in the 90s, Dante Chialvo. They claim that Turing mentioned something like this in the 50s. It's a little bit far-fetched, but you can read it like this if you want, some text by Turing. And of course, so this is called a second-order phase transition. You have a transition like this when it's continuous here. Of course, for the brain, what would be the control parameters? What would be the order parameters? In other words, if the brain is critical, what is the phase transition? So for the non-physicists, or perhaps even for some physicists, the favorite time model for physicists to understand the phase transition is ferromagnetism, in which so you have iron atoms in a crystal forming iron. If they try to align their magnetic moments, except that if temperature is too high, well, temperature wins, and they have disordered system, and nothing happens. You have, on average, zero magnetization. Half up, pointing up, half pointing down, and fluctuating a lot. If you have very low temperature, though they manage to align, and eventually one side won't win, and you have a macroscopic magnetization. That's the magnet that you put in your fridge, right? Boom. It's a magnet working. It's a magnet. If you increase the temperature, you have this scenario in which you have a critical point. So this is schematized here, having magnetization, macroscopic magnetization, as a function of temperature. If you increase temperature much, you reach a critical point, and then nothing happens from there on. And there's always a nice video on YouTube for anything you want. So this one is showing, what I want to show you now, is one particular kind of magnet whose critical temperature is low enough that you can reach it with a simple flame, okay? So, let me see if it works. Yeah, so this is a magnet, and there's another one. This is a magnet, which is very high. So it's above the critical temperature. Therefore, it is not a magnet right now, because the spins are not aligned. If you wait enough and the thing cools down, it crosses the critical temperature, it becomes a magnet, and you can see it becomes a magnet. There's also a spoiler version in which you know when things are going to happen. The thing happens first. We can wait 10 seconds, right? Let's say, there you go. Time 0, and this thing crosses the critical temperature, becomes a magnet. So you can see that, in case you didn't believe me, these things are real, okay? Critical phenomena and phase transitions. You have water to vapor, you have water to ice. Not all of these, actually, most of these are not second-order phase transition, most of them first-order phase transition, so let's not go into, this is why this is a favorite example, ferromagnetism. The question is, of course, why on earth would you expect that to be remotely true or even debatable, open for debate or worth bringing to the table? So it's a long story a little bit, but theory and models predicts that at criticality, one could have optimal transmission capacity. It's also debated in the very first paper on this field. Optimal information processing, there's a very nice paper on neurocomputation showing that a model that has learned some task, if you put in a critical point, somehow it learns best. Largest repertoire of metastable state because you have this transition between, so if you're in too high temperature, you have no stable states at all, you just have a mess. A mess is not much good for anything. And if you are low temperature, you have a very stable collective phenomenon. Well, very stable is not that good either because then it's very rigid. So the idea is if you are right in between, you have some maximum metastable state that you can work with. For instance, proxies for short-term memory kind of thing, right? And yeah, sure. I know. I doubt we would, because I'm not sure they are operating under optimal conditions, because they're operating under not the logic of the brain, right? Right now, so where are we in civilization today? So they have these large LLM models, right? ChatGPT and whatnot, and they're running on very large energy consumption because the training, well, the architecture is brain-inspired, but the training is not, right? So right now, people are trying to develop neuromorphic computing because the brain somehow solves this problem more efficiently than computer.
Way simpler, like the HDMI adapter in your laptop. It probably wants to optimally transmit information to your graphics card with a protection system, right? It needs a certain number of pixels and all these pictures, and does that with a few million transistors, I guess.
Yes, but not under natural conditions, right? The thing that is coming in is not exactly what organism faces. So the contexts are different. So I don't know what to expect, but it is under no obligation because it was man-made and stuff, for instance.
Claims are all with the big asterisk of if all the assumptions are met.
Yeah, always, yes. Yes, true.
Sense, critical makes more sense in a neural network context rather than a regular grid network as we find in microchips. That's my intuition, at least.
Yeah, well, yeah, and you can argue for the HDMI cable here, but perhaps not here and here and here. So there are many things together, and again, fair enough. What I'm giving here is a very hand-waving argument, okay? Just to start the conversation. And then I'll try to bring in slowly how this eventually becomes a testable prediction in the lab with non-anesthetized animals. This will be tomorrow, but we'll have a long way to there. Yeah, go ahead.
Hi, Professor. I was thinking if this is a feature of brains only, or we can describe biological organizations generally with this critical lens.
Yeah, be my guest. Yeah, if you want.
Okay.
Yeah. Perhaps, yeah. I mean, the idea, again, in a very hand-wavy fashion, is that these are all supposedly highly desirable features for brain. There will be some evolutionary pressure that will somehow make you be here. This is the hand-wave presentation of this idea. Okay? And if the brain is critical, what is the face of issue? This is a question that many people ask, including myself. And nowadays, I'm not so sure I'm really so concerned about this question, but I'm going to carry you through this idea using this question as an anchor. Okay? So at a critical point like this, theory in physics predicts precisely that you obtain a power law distribution for the size of an avalanche, decaying with a power law with some exponent t, a power law distribution for the duration with some exponent called tau t, and in the standard model for neuronal avalanche, since that experimental result in which they obtained tau equals 3/2 and tau t approximately equals 2, was that of a branch. Many physicists immediately identified those exponents as that of a branching process. Or if you want a fancy name for it, is a mean field rate percolation process. And what this is very simple, pictorially. Imagine this, each circle here is a neuron, and neurons are connected with some probability. So please notice, there's no inhibition here yet, right? Just excitatory neurons with some probability. And if probabilities are small, any activity you put here will eventually die out, and then you have silence. And then activity stops. This is called an absorbing state in theoretical physics, okay? So the game stops, that's it. If you have sufficiently high probability here, then this activity will eventually increase and saturate because of some refractory period or not, and then we have stable activity. So you see, you have one kind of activity, collective activity here, and another one here. Here you have silence, you have death. Here you have some stable stuff. And in between you have a critical point, exactly where you change from one to the other, right? So this is direct percolation, which is a model in physics which has like, I don't know, it's from the branching model is from the late 19th century, okay? It's a model to predict why some British surnames would survive and others wouldn't. That's a model, the Galton-Watson model. And guess what? The one who has more kids wins, right? So people have larger P's here, probabilities will win. And this model has a critical point. So this is well known since the late 19th century. And for this kind of model, therefore, you have a phase transition between an absorbing and an active phase. And here's another way of seeing this. This is density of active sites as a function of time. You start, say, with 10%. If you are here with small probabilities, we call subcritical, activity dies out very quickly. Here you have 3 runs. If you are supercritical, activity remains stable. If you are critical, you will eventually die out, but it takes longer, it fluctuates a lot. You don't know when it's going to happen. It is power law distribution. You don't have a characteristic time in which you're going to die, right? Again, this is just a toy model. And for this class of phenomenon, of model, these are the exponents which you can calculate analytically, and are the exponents that apparently were obtained in Dietmar Planck's lab. So this is why people got, oh, it looks like something I know. The physicists, okay? And there are also other properties I'm going to mention here, show in a minute. For instance, if you are at the critical point here, right here, suppose you stimulate this network a little bit. You put some external stimuli, like this, ta-ta-ta-ta-ta. And then if you are here, this ta-ta-ta-ta-ta will become some response of our network, which will then die, right? And this response is typically linear. The more you excite the network, the more it responds. Kind of boring. If you are here, right here, you get a nonlinear response. So H here will be this external field, and rho is the density. So actually, for this kind of class, universality class, in the jargon of physics, the exponent is 1/2. So the response is the square root of your stimulus here. And I'll show this in a minute, more clearly with a specific model. So this is MFDP will show up every now and then. It means mean field directed percolation. So this kind of model here. So an extremely simple model of a generic sensory system. And I know this immediately offends many biologists, because someone who works with audition and will be offended by me talking about this as being something remotely anything to do with audition or with olfaction or with vision or whatever. But just bear with me for a moment. Just let us assume it's a very simple model. It's a toy model. This is a model of a toy model of a neuron. It's like an excitable cellular automaton in which you are at rest here. If you are stimulated enough, then you spike, and then it becomes refractory for a number of times, and then you go back here and become susceptible for spiking again, right? And if you don't get stimulated, nothing happens. So imagine you take, this is the model I'm going to use here. And I put this, many of these guys connected in some topology, right? So here's one of these guys. I was one of them. 1, 2, 3, 4 is the other then. And I connected with probabilities. So this guy has 4 neighbors, and this local branching ratio is sigma I 1.7, which is the sum of these probabilities here, which means that on average, whenever this guy spikes here, let's assume it's, okay, this is a toy model of olfaction, okay? This is like your nose. So a molecule of vinegar goes by diffusion, reaches your nose, binds to one of your, or many receptors that will bind to it, and they will spike, right? And then they go to the olfactory bulb, and then they're connected with other neurons there. So this is what more or less somewhere in the olfactory bulb this will be happening. And so then it spikes here because it got it bound to a molecule, and then with some probability, this spike will be transmitted to others. And maybe to others, maybe not. It's probabilistic, just a simple model. And then this guy becomes refractory, and then everything stops. And then another stimulus comes, this time 2 neurons spiked and becomes refractory, and on, etc. Right? You got the picture, right? So this keeps going. Let's see what role criticality could play for sensory systems in this very offensively simple model, right? So what I'm showing here is a toy model of 50 by 50, that's 2,500 neurons in a square lattice, just because it's easier to show, right? Just to do topology, just because it's easier to show. And here I'm showing you in the horizontal axis in log scale the intensity of the stimulus, and in the vertical axis the intensity of the response. Here in linear scale, here in log scale. It's the same plot in 2 different scales. Okay? And let's see what happens if you have no coupling between neurons. So isolate the neurons in this very simple model. So what does it mean to have a weak stimulus? The stimulus here is just a Poisson rate at which these molecules hit your theoretical nose. Okay? So there you go. You have some, well, stimuli incoming and some neurospiking. What does it mean to increase stimulus intensity? Well, that happens hello? That happens more often. So, oops. And if you do a very strong stimulus, that happens even more often. I don't know what's going on. Hold on. There you go. You are saturated by your refractory period. So this is very boring, right? I mean, the more I stimulate, the more I get response. And so it won't be no surprise to anyone that this curve here, even though it's a log scale, you cannot see, it's a linear response, okay? So the slope is 1. Now, what happens if I introduce some coupling in the sense that I mentioned before? Some probability with, say, with first neighbors in this idea, in this simple topology here, right? What happens now is that you, as you introduce coupling, you have an increase of these responses here. Why? Because now you create this oops, where is it? Because now you create like these small excitable waves, right? Which amplify your response. So remember, if you are some neuron inside your head that's going to sense or interpret or make sense of this vinegar which you are sniffing, what you're going to receive is not this, it's this thing here, right? You get the response that these neurons in your peripheries are coding for somehow, right? So here you got a nice amplification of this weak signal here. So good, this is good, I think, I don't know. So these excitable waves mean you have amplification of your signal. And this amplification is irrelevant here in the high regime, the high stimulus regime, because here you are saturating anyway. Now, if you increase coupling even more, this amplification increases as well, if I only manage to show this. Oops, there you go. Right? So these excitable waves last longer, they propagate further away, and so this looks good. Looks good because now apparently, well, I have even more amplification. So you can, well, I keep increasing, right? So the more the better, that's what it would appear. Except, well, eventually you will reach what is called a critical coupling. So in a critical coupling, please note that the slope has changed, and I'm going to introduce a very, 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 very weak stimulus here. So you see that these waves now are lasting much longer. Actually, they are on the verge of never dying anymore. If I increase my coupling even more, and I'm going to argue in a minute why this is the optimal amplification. I'm going to argue for that in a second. If you increase even more your coupling, what happens? Well, you have a phase transition. Now, this very same weak coupling, you generate a wave, excitable wave, a stochastic wave, that never dies out. Now, this is a bad nose. Because what this means is that if this were my nose, someone opens a bottle of vinegar in the back of the room, one molecule by diffusion arrives here, and I smell vinegar for the rest of my life. Right? So this is not good. Now this is too much amplification. It's almost like taking the microphone, coming closer to this loudspeaker. If I do like this, very softly, and you can barely hear. If I come closer, 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 you can hear it eventually a little better because we have some reverberation. So this is a coupling. But if I come too close, everybody knows what happens. Audio feedback, right? And then you only hear the audio feedback. You won't hear anything that I'm tapping here. This is a bad coding, right? Bad sensor. So this supercritical excitable waves are too much amplification. I'm just arguing with my hands again. So you have this family of response curves in log scale, and I can show them like here. This is again, for some topology, from random graph topology, you can do these calculations analytically, and they mesh the simulations, blah, blah, blah. That's not really the point here. The point is that, well, here you have this critical exponent, 1/2, that you obtain right here, only at the critical point. And interestingly, this value of the response exponent agrees with many experimental results, agrees with Stevens' psychophysical exponent for brightness. So this is a psychophysical exponent. You put someone in the dark, let them adapt to the dark, and you show them bright spots of light whose intensity you control. So you control the physics, which is the horizontal axis. What is the intensity of the light that you're flashing at them? And the psychological part is the person reporting the intensity that he or she feels. Oh, this light is 2. This light is 7, and they normalize to 100 or something and take the average, and that's the experimental response. This is like, I don't know, 100 years old kind of result, psychophysics. So this exponent here agrees with the model only if the model is at the critical point, right? Otherwise, the exponent here is 1, here is 0. I don't know, doesn't even have an exponent here. If you take retinal ganglion cell responses, experimentally, this is, again, a linear log response is not good to compare with that, but if you take the same data and just put it in a log plot, you get some similar exponent. You take a detailed model of the retina. So Antonio Carlos Roque, our good friend, specialist in computational neuroscience, detailed model, conductance-based detailed computational neuroscience, you also get the same kind of non-trivial exponent if you use physiological values for the couplings and whatnot. Let me take a jump to 2013. So this group, led by Dante Calvo, took some connectome available of the human brain, DTI-based connectome, put a model inside, a very similar model to the one I showed you, and simulated this whole brain, simple brain. And then, once the model is working, you measure functional connectivity, which is like correlation between different sites, which is the only thing you can measure in the lab with fMRI, okay? And they compared the experimental results with the model results. And the response is that only when the model is at the critical point, you have a closer match with the experimental result of fMRI. If you're subcritical or supercritical in the model, the match is poor. The same year, Gustavo Deco published the same idea. So they have a connectome somehow here. They have some way more complicated neurodynamic model from which you obtain a functional connectivity, which you then compare with experimental data. So functional connectivity of the model with the data, and what is it that they obtain? That the fitting between empirical and simulated functional connectivity is maximum at the critical value of their model. So this thing comes back over and over, that you take these models and you tune it to the critical point, and somehow it fits your data best. The question is why? Why would you like to be here? Why would, see, in physics, that will be cheating, right? In physics, that will be cheating. I have a model that only works in one point, right? It doesn't work in any other point. In biology, well, do we have a good reason to be there? Because if we do, then natural selection kicks in. You have a bunch of homeostatic mechanisms at your disposition to do the job. So let me try to convince you that there will be a good reason to be there. I'm showing you the same family response curves now in linear log scale. And this is the definition used in the biology literature and the engineering literature a little bit as well, of dynamic range. So consider these points here as a generic response function, right? So here is some external stimuli. In my example, the intensity of some odorant, right? And here is this average spiking rate of the neurons, which is going to send the information further into the brain, downstream into the brain, right? So if you have, suppose you have this curve here. Suppose you have some 10 to the minus 5 stimulus here. You have this kind of response, ta, 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 ta. Now you increase to 10 to the minus 4, like 10 times. What is your response? Well, it's pretty much the same, right? So if you are a neuron in the brain, you cannot tell the difference between this and this, because you have like a plateau here. So plateau is bad, because it's poorly invertible, right? You cannot read what was here by looking at this. So this very crude definition of dynamic range employed in biology is that you take a sigmoid curve like this, which is very generic, and you discount 10%, well, you take this interval between maximum and minimum, you discount 10% above this plateau and 10% below the upper plateau, just because this is completely arbitrary, 10%, it could be 5%, it doesn't matter. Some percent, you remove that part because you don't like it, because it's poor coding, and whatever is left here is considered good coding, you can invert somehow, and the range of stimuli, which is covered by this good portion of the curve, is called the dynamic range. So in this case, you have 2.2 decades, which is 22 decibels, of stimulus intensity that this curve will be coding well. Okay? It's okay? That's a operational definition, not mine. I didn't invent this. This is in the literature. People use this kind of measurement. If you're thinking, well, can't you do better with information theory? Yes, I know, of course, it can be done, okay? But this is simpler. So consider what happens in the model. In the model, you have here this maximum response, which is more or less always the same. You have this upper plateau, 10% below it, it's somewhere here. And now consider this supercritical coupling here. So for each curve, I have a different coupling. So zero coupling, weak coupling, less weak, stronger, 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 stronger coupling, right? So for this zero coupling, this is my cutoff for the 10% here, and therefore, this is my dynamic range. Graphically showing, you can calculate the numbers. It's like less than 20 decibels. And then once I increase the coupling, this curve shifts to the left, where this one doesn't change much. Therefore, your dynamic range increases. If I keep increasing the coupling, dynamic range of stimuli that are well coded by this function increases, and increases, and increases, and increases as you reach a critical coupling. Now, if you go further, what happens? Audio feedback, right? You have all of a sudden this self-sustained activity that you have to discount, because now anything here won't be coded well, because you have your network working on itself. It's not caring for what you're inputting. So once you discount for that, your dynamic range is like this. And everybody who has ever tried had a problem with audio feedback, either singing in a karaoke or at home or playing. If you increase your volume even more, it gets worse, right? If you keep increasing the coupling here, it gets worse. Your dynamic range decreases, and decreases, and decreases, and decreases. And therefore, as a function of coupling, this dynamic range initially increases, reaches a maximum at the critical point, and then decreases, right? So if you make a plot of one against the other, what you're going to have is something like this. So dynamic range measured in decibels as a function of coupling has a peak of, at criticality. And therefore, if we assume or agree that having good dynamic range, being able to code for a large range of stimuli is a good thing, then you would have a good reason to be here, because here you'd maximize dynamic range and would explain how you obtain those exponents that you observe experimentally. Okay? So this is the rationale. It will be 20 years now, this paper, Jesus. So actually, so this is very generic. We did for random graphs for different topologies, different kinds of model, blah, 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 blah. So this has been explored ad nauseum, and it's pretty much, I don't know if there's any model left to be done in that respect. And one particular thing that I liked, which was developed by Osami Kinauchi, my good friend and collaborator, is that you can even play with this model, introduce homeostatic mechanisms, so dynamics in the connections, so that you don't even have to tune your model here. You put, this is sigma, this is the coupling here. You can tune your model, you don't have to tune, you put whatever you want, and the model self-tunes to the critical point. So you can make models of self-organized critical point, right? So this can be done for very simple models, again, we're talking about, right? Yeah?
Question, so this argument based on the dynamic range, I wonder if it, like in the sensory part of the brain, it's always the case that we want to maximize the dynamic range. So
I'm not sure it's always the case. So of course the devil is in the details, right? I didn't mention that, but I can mention it in full honesty. This is what is called rate coding. It's just assuming that you're coding with the mean fire rate of a population of neurons, which we know is not always the case. So there, my model goes away immediately, for instance, right? And in some cases, you don't want large dynamic range, you want small dynamic range. So there's a whole complication if you go into the details, right? So I'm just introducing a very general idea that why this might be a good idea. Of course, it would be nice if it were true, right? It's just a model. And some people have tried it. So again, in Dietmar Plan's lab, I was very happy they tried these ideas. And again, for cortical slices, they stimulate the slices and they control for excitation and inhibition, playing with drugs that either block excitation or inhibition. And the no drug condition is usually closer to the peak of the dynamic range. If you disturb it, the dynamic range decreases to either side. So it was I was very happy for about 1 year when this paper came out, because okay, someone bothered to test these ideas and it kind of worked. Again, in a cortical slice. And also they tried for anesthetized rats in somatosensory cortex, stimulating the whiskers. And more or less it worked, right? You have some kind of peak, dirty one, but still. But we were happy because some very, very simple ideas from a very, 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 very simple model somehow rendered some ideas that could be tested and gave some insight. Okay, cool. So this is the scenario that more or less got established for many years, and for some people it's still established. People try to essentially start to identify with these exponents as some magical numbers that data should obey. However, things are not that simple, obviously, right? So let's start to complicate matters and throw some sand in the fan. So, experimentally, the avalanche exponents are not always the ones I showed you. And so the first experiments I showed were in 2003. By 2015, you have Woody Shu, formerly a postdoc in Dietmar Plan's lab, and then already a PI in the US, doing experiments with the ex vivo visual cortex of the turtle. So they extract the whole brain of the turtle, and they stimulate the retina with some movies and measure activity in its visual cortex. And they observe this kind of avalanches, and they do the statistics, and they obtain the exponents, okay? They do everything again. What's the probability of showing an avalanche of size S? Oh, S to the power minus tau. What's the probability of obtaining an avalanche of duration T? Well, it turns out to be T to the power minus T tau, tau T. So what are the values of these exponents? According to the model that I showed you, which is very convenient for me, for my model, for instance, well, actually, you don't have a single value of these exponents. You have a family of values. And tau T equals 2 is here, and tau equals 3/2 is not even in the scale of the plot. You have to increase the plot like this and put here. So the model I showed you is here. This is the model, the standard model of this field, and these are the data. Not very good, right? So this is kind of, okay, this is starting to get...
But the avalanches of the data always follow a power law.
This turns out to be an issue as well, in the sense that these are not any power laws. These are power law distributions, right? One thing is to go to a lab in physics and calculate the frequency of the pendulum as square root of G over L, the length of the pendulum, and then make a plot log here and log here, and obtain 1/1/2. This is kind of power law that can use least squares to fit, for instance, and that's fine. Power law distribution has to be fit with more statistically correct methods, like maximum likelihood estimator and whatnot. You have always a cutoff, so this cutoff is an issue. You have a lower cutoff, this is also an issue. So to the extent that all these difficulties have been circumvented, yes. But you could argue whether they have really been circumvented. So this is an issue of the field. And I'm going to come back to that. So this is a methodological vulnerability of this kind of approach. which I'm going to abandon tomorrow. So, but today I have to extinguish this thing because this has been the history of the field for almost 20 years. Yeah.
So based on this great question, can you show us the raw data that went into this? Like how the software different?
No,
And probably this is much later than the first experiment, so this is probably a lot more electrodes, right, in recording sites, so they can really see the differences in slopes.
Not really. Not many electrodes more. Let me show. I think these MEAs also have still on the orders back in the day, or for this setup at least. It's not my setup, okay? So I don't know. But I think it's in the orders of tens of electrodes. What?
These are not 28 examples, they might be 28 electrodes. Based on the y-axis.
Say that again? I'm sorry?
How many electrodes do we see?
Oh, I forgot. This, what?
In my experiments, my electrodes have about 10,000 recording sites.
Yeah, no. Back in the day, they were way less than that. I mean, this is not my experiment, so I don't remember anymore. But certainly not in thousands, in the 1,000 range. Today we can do this. Yes, definitely, yes. So the neuropixels nowadays are changing this. Well, and two-photon microscopy, right? So people are just forgetting about electrophysiology and just going to the microscope. And it's a little bit worse than that, actually. Even if you had many electrons. I'm going to show you some. It's still today. So I'm in the part of the talk where things are going to get bad, okay? So this is a bad result for people who believed in this kind of model. I'm not one of them, by the way. I did use the model, but, okay? It's just a model. So this is the model. And there are some previous results suggesting an intriguing level dependence on power distributions and the level of synchronization. Let me show you very briefly some kind of very old experiments. This is data from Siddhartha Ribeiro back in the time when he was in Nicolelli's lab. So this is like more than 20-year-old result. Then really is like 30 electrodes or something like that, tungsten electrodes. And so this is a raster plot of primary visual cortex of an awake rat, like this, and you see that it's not very synchronous, whether you take the same kind of setup, same electrodes, and you anesthetize with a deep dose of ketamine xylazine, you have this the synchronized activity. And you can try to do avalanches and see what you get. What you get is something like this. So essentially, this is one kind of anesthesia that you can recover from. You can come back, you can anesthetize the animal and then let it recover and wake up. So every curve here is every, 30 minutes or so. When it's deeply anesthetized, you have some kind of power law, again with a huge cutoff here, some kind of power law. And as the animal awakes from here to here, these power laws get twisted and turned until they become nothing like a power law. This is not a power law, okay? The upper curves, they are not power laws. I wouldn't claim they were power laws. Actually, physicists in the 90s, when self-organized criticality was on the hype, they would fit power laws no matter what, right? The points like this, just pass a straight line like this. So we're not doing this. Actually, this is best fit by log normals, stuff like this. So this is kind of complicated because my model doesn't say anything about oscillations. Remember, it's a phase transition between silence and an active state. And these apparent some kind of oscillation here. That's strange. Okay, so there's a conundrum here, right? And the exponents are nothing like 1.5. They could be anything here. I don't know. So if this looks confusing to you, welcome to our world, because we were confused for a long time. Many people were confused for a long time, not knowing very well what to do with this thing. And would it still make sense to talk about this standard model to explain the phenomenon? So let me briefly talk about cortical states, which is a field it's well established in neuroscience, and let me walk you through this idea. So this is a very nice review. in which they define cortical states, they motivate the idea. So the brain continuously adapts its processing machinery to behavioral demands. To achieve this, it rapidly modulates the operating mode of cortical circuits, controlling the way that information is transformed and routed. Experimentally, the definition of state in biology, in neuroscience, differs from the definition of state in physics. This will be more like a thermodynamic state. So the dynamics of network activity on a time scale of seconds, okay? So it's not state in a sense of classical mechanics, right? The point is, but more collective dynamics, like is it liquid or gas? And for instance, you have this synchronized state, roughly put it like this, which is what you observe when the animal is drowsy, quiescent, on slow wave sleep. And you have desynchronized state when the animal is actively behaving, alert. And this can be characterized in a number of ways. For instance, you have low frequency LFP fluctuation here, if you look at the LFP, whereas here you have less low frequency LFP fluctuations. You have correlated spike in activity. Can you see the correlation between the three different neurons here? Whereas here, and if you calculate the pairwise correlation between all the neurons and do a histogram of their pairwise correlation, so I have n neurons. I have n minus 1 divided by 2 pairs of neurons. I calculate the correlations between all these pairs and do the histogram. So what you will obtain here is some histogram with some probability, some positive average here, right? Whereas here, you have uncorrelated spike in activity in the sense that you have some positively correlated, some negatively, and the average is close to 0, more or less. Okay? So this experimental data, people know that this happens when the animal is doing this, which is nothing, right? Kind of sleeping, quiet, and people know that this is what will be observed in the cortex while the animal is alert. Actually, cortical state is not bimodal. It's not like synchronized and desynchronized. There's a continuum of states in between. And since we had these results of confusing signatures of criticality with oscillations, without oscillations, we asked ourselves, well, could we perhaps exploit this continuum that exists in the brain to try to solve this conundrum between synchronization and criticality? So this must have been 2017 or 18 when we tried to start to develop these ideas. And the question, how can you do it under some kind of control? We then use this solution, which is urethane anesthesia. It's a kind of anesthesia that does the following. Let me show you what urethane does. So this is, I'm going to tell the story today of 2 papers separated by 2 years that contradict each other, okay? So I like to do this. I have no problem in shooting my own foot if need be. So what happens here is the following. So let me tell about this paper which was led by Antonio Fontanelli, nowadays working in Woody Schu's lab. And Nivaldo Vasconcelos is a PI in the biomedical engineering in our university. So we took this silicon probe measurements. I think these were still 32-channel silicon probes, and implanted in primary visual cortex of the rat under urethane anesthesia. If you do this, what do you see? This kind of anesthesia. It's a very stable anesthesia, okay? It can last for hours. This is like 2 to 3 hour recordings. And what I'm plotting here is the coefficient of variation, henceforth called by its nickname, CV. It's the CV is just a standard deviation divided by the average of the population firing rate. Let me show you what this means. So this purple here amounts to this purple here. So it's a very small value of coefficient of variation of the population firing rate, which is this purple curve here, which corresponds to this raster plot here. So if you look at this, I think you would agree with me that this will be called desynchronized activity. You have a bunch of spikes. It doesn't look like they're synchronized. and these fluctuations are small. See, if the fluctuations are small, the standard deviation of this thing is small, and therefore, when you divide it by the mean, it gives you a small value. This is why CV has a small number here, right? Okay? Take this other point here. Here, large CV, you have this and then silence, then and then silence, and then silence, and then so it is very highly synchronized state. So urethane does that. That's what it's known for. It's kinds of goes around between very desynchronized and very synchronized states. So it's a way to explore these different regimes. And you have things in between, like this one, which is intermediate value of CV, like here, okay? And you cannot control this. It does it on its own. Each animal has its own history. We don't know what's going to happen, right? So what did you do? Well, we try to parse these, do the avalanche stuff, and parse these results according to CV. Except that, okay, so we're going to try to do spike avalanches here, calculating everything between silence. Of course, this depends on the bin. It's a long story about the bin. And we have some power laws which are nice, some which are ugly, so this is the limit of power laws not being valid anymore. The different colors here mean different CV choices. We vary CV continuously with, I don't know, half a dozen animals. And we extracted the exponents from the red curve here, which doesn't agree with 1.5, and the exponent here, which doesn't agree with 2 necessarily. And what we obtain is that exponents change continuously with CV, which is very bad news for physicists, because exponents should have a definite value in a phase transition. That's what you learn in statistical mechanics course. So what do we do with that? Is there a criterion to choose which are the true ones, if that question makes any sense at all in the first place? And actually, yes, or so we thought. Let me go through this. We're talking about 2019 now, many, many years before the first exponent. By then, people had realized that statistical mechanics had other tools to offer beyond just looking at this two power law distribution. As it turns out, that if the system is critical, the average size should scale with lifetime with another exponent, like here, and this is true in our data. Not only that, if this is true, and this is true, and the system is critical, these exponents are not independent, and they should obey a scaling relation between the exponents. Again, statistical mechanics. And note that the two sides of these equations are independently evaluated from experimental data and vary with the level of synchronization of the data. So this comes from this plot, this comes from these other plots. So the question was, we asked, is the scaling relation satisfied somehow as this level of variability changes? And the answer is yes. So I'm plotting the right-hand side on one plot and the left-hand side on the other one, and they match here. So there's an intermediate CV value for which you have the conditions for a critical point. It's in a much more, it's a higher theoretical bar to pass to have this power law, this power law, this power law, and this scale relation satisfied at the same time, okay? So we were very happy that this was true, and we asked, and not only that, the residence time has a peak right around the critical point for these anesthetized animals. We also got data from the internet that were freely available and verified the same kind of crossing for different setups. And since we have a critical point, what are the exponents here? Are they compatible with this model? And the answer was, these are all the exponent plane that you obtain for this data anesthetized rats are here. And the exponents do not agree very well with that model. The exponent for freely moving mice that we got from the freely available data do not agree with the model. Actually, the black dot here is mean field errata percolation. And we get data from the turtle that I mentioned before and put it here, and everything falls into the same line here, very strangely. And to this day, we do not understand this, why. okay? But anyway, so the exponents are not mean field speculation. They seem to require a different model. That was exciting for us because they will suggest, okay, we need a new model. We don't know what's going on. This is good. Yeah.
So I have a question because the whole argument goes around that criticality is important for the brain because it's optimal in a sensing or cognitive way.
Exactly.
Anesthetized rats are not optimal in any cognitive way.
Exactly. Thank you. Perfect. That's perfect. Okay. I agree. So the rationale that has been used that, okay, the animal, if it works on a critical point, it will have the mechanisms to take you there homeostatically, right? It cannot self-balance. And you should be able to see this even in an anesthetized animal. So this is the point that has been holding people doing the experiments that they're doing. But I promise you that the last thing I'm going to show you tomorrow is a signature of criticality of an awake animal performing a task and showing signs of criticality when it performs best. Very huge spoiler. But so I'm getting there. But your question is the elephant in the room of the field. In order to measure things well, it's kind of Heisenberg principle of biology, right? In order, if you want to measure well, you have to anesthetize the animal. But then it's not doing anything. And your argument is that it is optimal, whatever you're measuring is good for the behavior, which you're eliminating by construction. So yes.
Thank you.
I'm running out of time, I'm sorry. So the main message here of that paper, but okay, we have a very theoretical physics high bar obeyed by this data, which is the strongest signature of criticality that you could have by that day, and doesn't agree with the toy model, with the standard model. So we need a new model. This we like in physics. A model being wrong is good because we need new models. Then we can play, right? And there's a cross model here. It is a model which has some oscillations and which seem to satisfy the data here. So this could be a hope. So this result here wasn't bought by one of my students. So in my own group, I had a student who didn't quite believe. He didn't buy what we were saying. I don't think so. So to my despair and pride, he decided to challenge us and show that what we were seeing were just a bogus phenomenon. And this is what I'm going to end with today. I'm not going to talk about the third one today. It's tomorrow. So what did Tawan do? So he did the following. Let me take a mean field articulation model that I know belongs to this class. The exponents are classic. The model is trivial in that sense. And I'm going to subject this trivial model to constraints that the data are subjected to. For instance, there is severe subsampling. This is the question that he asked me. The number of electrodes, like tens, hundreds of electrodes. It's nothing compared to the neurons which are involved in that region, even a small one, right? So there's severe subsampling, spatial subsampling of your activity from which you are deriving things and making conclusions. And do the same thing. So I won't go into the details. This is a very simple model with some kind of stochastic integrated fire, with some inhibition. So here we have inhibition, by the way. And so inhibition G, just look at G. As increased G, your activity goes down, eventually dies out. So here we have an absorbing state phase transition like the one I showed before, except with a more complicated model. It doesn't matter. And if you, to prove that you are in this class, you calculate, this is the model in the supercritical, critical, supercritical regime, okay, here. And the power laws in the model now are much more beautiful than the data. Of course, I can have 2, 3, 4 decades, as many as you want. Just increase the sim. It's 10 to the 5 neurons you can simulate very easily on your laptop, okay? And tau equals 1.5, tau t equals 2, 1 over sigma nu z, that our exponent, equals 2. So everything is perfect, the model is perfect, everything works as it should. It is mean field direct calculation. Fine. But I'm here, I'm sampling everybody, all the 10 to the 5 neurons. If this were a rat, which it is not, but if it were a rat, I would be sampling not all 10 to the 5 neurons, but I have like, even with the silicon probes, I don't know, hundreds of neurons, tops, with 32 electrons or 64, I don't know. And so if I sample 100 out of 10 to the 5 neurons, what do I get? This is Tau 1's results. This is the model, and first, you see that you have this CV as a function of time, which does not like behavior like the data, but you do have a wide CV distribution, even for a fixed parameter here. Each color is a different control parameter. So CV, which we were interpreting before in the experimental data as a control parameter, according to this model, is not a very good control parameter. We were wrong in that, if the model is right. And here, if you have, but changing just a little bit about the critical point. The critical point is G equals 1.5. You change just a little bit around the critical point, you can have asynchronous behavior, synchronous behavior. You can have power loss, poor power loss again, because now you're subsampling, your data is poor. But with exponents that more or less match what you see experimentally. This is data, this is the model. The only thing that the model does not reproduce is this time scale, this time series of CV, which is much richer here than it is here. But the other things are quite fine. In particular, to my despair and pride, I insist, by my own PhD student decided to defy us, the scale relation in which you have one exponent and the combination of exponents, this experimental data, with the spread of exponents that I shown you before, and this is what the model does. So what the model is doing is that, look at here, this is the exponent for a fully sampled model. These are the exponents that you obtain by just fitting the data of a subsample data. The subsampling completely distorts your results and changes everything and still gives you some kind of signal of criticality, which is crossing here. So this model pretty much destroyed our previous results, or our previous claims in a way, in the sense that actually it is not true that you need a different model to get those results. A very trivial model can do it here. Moreover, it's even more important than that, perhaps, on a more positive note, blah, 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 the values are, again, not mean field calculation, and they're compatible within error bar. So this very simple model with subsampling explain the data. That's it. Or that kind of data, right? Again, the spread of exponents that you see in the data and in the model only work if you're very close to criticality. 3% of criticality you don't see anymore. See, you have this bogus thing that you don't see in the data, but you see in the model if you're more than 3% away from the critical point. So this very small range of coupling values gives a very large range of CV values. And this is a control parameter, a good one because I built the model from scratch. So if we had before this interpretation from the data of this residence time that you are critical here where the crossing happens, which is the maximum residence time, but it's still en passant, right? You're going from here to here to here to here. You're not always critical here. You're far away from criticality a bunch of the time. This model changes everything. What the model is saying that actually this axis that you're referring to as a control parameter, it is not. The control parameter is here. This wide range of CVs is actually obtained by a very small range of a true control parameter. Therefore, according to the model, it is not true that we are a criticality en passant, but rather that the data, the model suggests that you are all the time near the critical point, which is an interesting result. So we got confused about the experimental results, and maybe this is true. And in this sense, the model also suggests that these highly synchronous and highly asynchronous states are just fluctuations around the critical point, which is a very bold interpretation of asynchronous and synchronous states. So these were the conclusions, which I'm going to skip because of the time. And just to finish this very brief first passage on criticality, so people focus most of their effort these avalanche statistics, which, even though it has a solid theoretical background, it has fitting issues, like related questions. Really, it's trickier than you think. It doesn't look tricky, but I promise it is, right? You change a little bit your parameters, the points change your conclusions. This is tricky. And like, as I've just shown you, these results are strongly distorted by subsampling, especially in spikes. I understand you talked about LFP and spikes. So LFP are more robust. Spikes are supposed to be richer, but they're more fragile because they have only local. You miss a lot of spikes. You don't get them all. And just to mention, there are other tools in this toolkit. For instance, long-range time correlations as measured by determinate fluctuation analysis was actually the first signature of brain criticality in the whole brain in EEG data by Licklinger Hansen in 2001. And there's a model which kind of mimics this behavior here. Very interesting paper, Journal of Neuroscience 2012. And more recently, Bill Bialek, good friend of ours, good friend of this place, introduced the idea of phenomenological normalization group, which, even though he didn't build it for that purpose, it's been very useful. It's very useful for that purpose. So my idea is that I'm going to talk about this thing tomorrow. Okay? So tomorrow will be about this new technique when I will eventually reach this third paper, and another one to answer his question about behavior. We're still talking about anesthesia here, right? Anesthesia, anesthetize animals. So your question, thank you for your question. It's a very important one. Let's not forget that the whole idea in the beginning, the whole promise, was that this thing was to give you functional advantages somehow, right? And for the moment, unless you believe that the dynamic range of a slice is a good thing, which they have shown experimentally, well, it's just a slice, right? We haven't shown much. So I'll come back to that. Danielle had asked me to give you references, and so I put in the Google Drives these 4 papers. If you don't have, if you're lazy, just read the first one. The first one is just a classic, okay? If you don't, raise your hand if you know this paper. Do you know this paper? Okay, those who don't, please read it. It's very simple. There's no calculations. This was published in 1972 by P.W. Anderson, a couple of years before he got awarded the Nobel Prize in Physics. And he just argues for how collective phenomena are a different thing than the original phenomena of their individual parts. But he argues very beautifully and very timely back in the day when particle physicists claimed that they had the most profound truth of everything because they had the basic science. Well, but what does that change for the economy, for sociology, for chemistry, etc.? So if you have to read just one paper, just read that one. It's spectacular, okay? It's there. Thank you. I'm sorry for the time. Questions, please.
I have a question. So you were talking about this underlying functional advantage that, like, evolution would have found. Do you know other similar results in other species, or like C. elegans, or species there is easier to maybe, or like flies that we know a lot about the
Well, you have now these impressive results with those, what was it again? Larvae, which they could image like all the neurons, right? And right. So yes, I think you just gave me an idea of a slide that I'm missing for tomorrow. So today I presented these neuronal avalanches, the idea of criticality, and I hope I've shown you that these avalanches have methodological issues, right? And so people have come up with other ideas. One of them is the one I want to present tomorrow, which by Bill Bialik's group on normalization group. So an Australian group, forgot the name of the first author, sorry, just took all the available data, multi-neuron data, from different species, from, I think it's zebrafish larvae or something, all the way to humans, and ran this kind of analysis in search for signatures of scale, non-trivial scaling and criticality, and they found them in all of them. So I think that's the best answer I can give it to you. And C. elegans is tough because C. elegans has just, I don't know, 300 years or something, right? Then you're on the verge of not being able to define criticality properly in physics, but that's a good question. I'm not sure they look at C. elegans, for instance. But so they have been looking across species, yes. So most of the results are for whenever they are invasive, mostly rats, rodents, and the non-invasive results are for humans. I didn't mention many stuff, but so fMRI, which has excellent spatial resolution and many voxels, so many sites, also show very strong signatures of criticality. But I'm going to show these results in this other technique. I think they're, if you believe the technique, they're more convincing.
So can I do a follow-up question?
Yeah, sure.
And what about like when people don't find signs of criticality? So like examples that people tried but there isn't.
Yeah.
Are there like a lot? So, and I was thinking like this could also be very informative of when criticality happens.
Yes, I think so. I think the answer to this question has changed historically, because in the beginning, people are doing avalanches with small number of electrodes. You could always blame the small number of electrodes and keep the hopes that you're not seen because you're not looking well enough, the right technique. And nowadays, these things are getting more refined, I think, and people are starting to understand that, well, it's not everything that needs to be critical. The brain, there's no such a thing as the brain in the sense that if you're performing a visual test, your visual cortex and thalamus and whatnot is going to be involved, your retina, but not your auditory cortex necessarily, right? So why would the auditory cortex be critical? So people are trying to refine these ideas as data becomes more abundant and the techniques become better. And I don't know if that's what you're asking, but I do see religious people in the field that try to find it however they can, and if they don't find it, they claim something. But there are people who understand that it's not always necessarily the case, and why that is so. Well, again, so to go back to my first slide, the golf course and the forest, initially it was just a guiding idea. So is this idea helpful somehow to guide research? And it has given some fruits, I think. And maybe we are now in a position where we can refine the question. Well, yes, it is useful to a certain extent, to this extent and given those conditions. And I hope that tomorrow I'm going to show you some results. So what I'm extending my promises again, tomorrow I'm going to show not only that this can happen in a behaving animal, but this criticality idea is context dependent, to borrow an expression from Asaf's talk today. So in some contexts, it appears, some contexts it doesn't, which to me is amazing and well, gratifying and still perplexing, because it posits a major challenge for modeling, for instance, right? But again, I'm super interpreting perhaps your question in the sense that, but I've seen religious people, and I know we have a philosopher in the room, right? Who is a philosopher again? You have a philosopher or something. Some people have compared this idea of criticality to Platonism, in the sense that you idealize, you idealize something, and you hope that you project this thing always into this direction, which would be harmful, I think, and not very, very scientific. But as a guiding principle to check make some questions, I think it's been doing well in provoking us questions and trying to answer them. I think it's been fruitful, I think.
I also have a question relative to one of the early examples with the olfactory system, and where you chatted about the optimality of a slope of 1/2 for the De Vries law for vision and things like that. I can see why this is a good solution, but only for a certain set of statistical features of the input.
Right.
So for many other examples, sensory systems are often tuned for like optimal information transmission, and so the sensors or the tuning curves have properties that reflect the statistical structures of the inputs that the animals
Exactly.
like in
Yes.
the fruit fly, there's a common example with the wind direction system and things like that. And I was wondering how you can link these two domains, because the examples that you present here were very carefully chosen, that they would have linear relationship, not monotonous relationships between the response and the input. But that is not that can't universally hold, because stimuli are very complicated and particular statistical ratios, particularly in the visual world. So how does that change then?
Yeah. Well, that would change things completely. And so the model obviously is it's a toy model, right? Of a very simple one, framed in a way that the statistical feature that we're looking at, so intensity, is very, very crude, right? And so a crude model for a crude measurement. So they worked out all right. And for a more sophisticated statistical feature, you need a more sophisticated model, and they are, I think, around for some modalities and stuff like this to some extent. And people in this community just haven't looked at them, so I don't know the answer. So the models that we have looked at, say the Pottius-Dismus model of a cortical column, very complicated model, etc. And you can find signal criticality there. What does that mean? Well, I don't know. It's just one general cortical column, right? But I'm in a tough spot here because I showed some very simple model that worked, but people haven't been working on the data because they haven't been able to work on models that do what needs to be done. So I would be too daring, perhaps, if I could promise you that, well, someone will come up with a model that does the job. I'm not sure of that. So I don't know. But what people, so just to go back to something you mentioned, that people have looked at the statistics of natural images in natural conditions, and these are scale-free, they have scale-free statistics very often, right? So the world, not this one, I mean here, but out in the forest where we come from, this one seems to have scale-free statistics. So this is another reason why, well, maybe a scale-free system will be better suited to deal with that. So another hand-waving argument, not to answer your question in detail, but in a hand-wavy form, which is what I'm doing most of my day, right? I don't know.
Okay, the thing that you are trying to do is to test the criticality of this collection of neurons, if I'm understanding well. The thing that I want to, or that I'm wondering is if the criticality of the global structure of the brain could help to establish this criticality. I mean, what Bruno Mota showed us on Monday or on Tuesday is that the brain has a self-similarity, and it is an characteristic of a critical of criticality, yes, of a critical state. Then, if those two things could be related, is what I.
Yeah, this is a big jump. I have no idea. I've been teasing Bruno for a while because he's a structural guy, right? He does structure, anatomy, and we do model dynamics and data. So the distance is very large. The answer is, I don't know. I would love to say that I guess so, but even that would be too much. I'm not sure. I just don't know. Maybe Bruno has some insight to offer, yeah.
Well, I actually think that those two types of scale freeness may be completely unrelated because they may arise from very different reasons.
Yeah.
My guess would be that. But certainly the fact that you have a fractal brain must have some reflection on the connectomics of that brain. But to the same thing more definite, you really have to have a connectomics map across white matter because lots of connections are local and those are not very much affected by gerification. So that's my impression generally. Now, I actually have a question for you. So the fact that you're in a critical state implies that there exists a subcritical and supercritical state. And I mean, if I had to guess, I would imagine there are states in the brain such as an epileptic seizure or a coma that might be characterized by either subcritical or supercritical. And have you tried to characterize abnormal states that can be characterized as either subcritical or supercritical?
Well, I haven't, but people has. So there's there are recent, well, a couple of years old, review entitled something like "Why Criticality is Clinically Relevant." So they try to associate these markers, they're different markers, with health and disease to some extent. So it's a new field. So yeah, people have been trying that. Well, if I go back to this long-range time correlation that I mentioned, so DFA analysis, determinate fluctuation analysis, well, this was introduced in '95, right? And the first data set they used was heartbeats. So if you think of a heart, what is your favorite toy model of a heart? Well, it's a limit cycle, right? Like ta, 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 ta. It's a periodic system. Well, you think, well, it's not really a periodic system because it has some noise. Okay, so it's a periodic system with plus some white noise, right? So ta, 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 with some fluctuations, ta, 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 right? You would think this is a reasonable toy model of the heart. Well, if you measure your heart and this means that you have no long-range fluctuations, and that means you're sick. You should go see a cardiologist because healthy hearts have long-range time correlations in their fluctuations. This is experimental result, again, in this direction. Of course, it's much simpler, it's just a heart, right? But so people have been trying to go there.
But that's a beautiful result. That's one of the things I had in mind when I asked the question.
Yeah, so people are trying to do this. Well, with EEG data and which is very noisy and dirty and stuff like this. But so yes, people have been trying to do this. And also related to consciousness, which is, well, the hardest question of all, right? And yeah, people are trying. I can show this review if you want, yeah.
Okay, let's thank Mauro.
Thank you.
So now we'll have the coffee break.